the exact same genetic mutation, but only one of them actually develops a disease? It's honestly one of the core mysteries of modern genetics. You see, just having the DNA instruction manual isn't enough, especially if we don't understand how the cell is actually reading it.
Welcome to today's Explainer. I am incredibly excited to share this one with you because we're looking at a massive computational leap that is, quite literally, changing the future of medicine. Today, we're breaking down a groundbreaking study on a revolutionary tool called EpiXcan, and we'll see exactly how it's decoding disease genetics like never before.
All right, here's our roadmap for today. First, decoding the genetics mystery. Second, the epigenetic upgrade, EpiXcan. Third, outperforming previous prediction models. Fourth, mapping traits to specific tissues. And finally, repurposing drugs for complex diseases.
Let's jump right in with part 1, decoding the genetics mystery.
To figure out how our DNA connects to our health, scientists rely on something called transcriptome-wide association studies, or TWAS for short. Think of TWAS as our foundational tool. It's the baseline method used to integrate gene expression data with genetic risk variations. Basically, it's how researchers try to link a specific mutation to a specific physical trait or a disease. But historically, well, these prediction studies have hit a bit of a wall when it comes to accuracy.
Let's break it down like this. Imagine your DNA is the text of a massive, super complicated book. Epigenetics, which are the chemical modifications around your DNA, they're basically the highlighters and margin notes, telling the cell exactly what to read, what to completely ignore, and what's highly important. The problem is, older prediction methods only looked at the raw, linear text. They completely missed those crucial epigenetic sticky notes that actually control the whole system. Which brings us to part 2, the epigenetic upgrade, EpiX.
So how does this whole thing actually work? EpiXcan uses 3 pretty brilliant steps. First up, it estimates probabilities using those epigenetic clues. It's essentially calculating how likely a specific genetic variation is to have a regulatory role based on those biological highlighter marks. Second, it rescales those probabilities into penalty factors via adaptive mapping. And third, it runs all that data through an advanced predictive model. By actually factoring in those epigenetic highlights, EpiXcan can accurately predict exactly which genes are getting turned on or off.
Let's look at part 3, outperforming previous prediction models.
Okay, the crucial point here really comes down to this comparison. EpiXcan was the older, widely used method, and it predicted gene expression using only the linear genetic text. EpiXcan, on the other hand, is the upgraded method. By integrating that epigenetic data, the actual biological context of the genome, it vastly improves predictive accuracy. When researchers ran the numbers across 58 different complex traits, EpiXcan absolutely left the old EpiXcan model in the dust. I mean, just look at this number, 8.47%. Now, in the world of complex genetic statistics, this is a massive boost in predictive power. Using epigenetics effectively gives scientists an 8.47% increase in their effective sample size. That means they get the statistical power of having significantly more patients in a study without actually needing to recruit a single new person. It's a real game changer.
And you can totally see this tech upgrade in action right here. Notice how the blue EpiXcan bars completely dominate the older gold bars across the board. It doesn't matter if we're looking at brain tissue, the aorta, or subcutaneous fat. EpiXcan detects almost 10% more significant associations overall. But it's not just about finding more of the same stuff. It's about finding what was literally invisible before. Because EpiXcan is so much more sensitive, it uncovers these unique hidden targets. In fact, it discovered nearly 80% more unique genes than older models. Just as an example, it identified the SD gene, which it powerfully linked to waist-adjusted BMI. Why does that matter to us? Well, researchers already know that knocking out the SD gene in mice actively reduces diet-induced weight gain and body adiposity. So EpiXcan pinpointed a major obesity target that older methods completely overlooked.
Moving on to part 4, mapping traits to specific tissues.
Taking a look at these numbers proves just how remarkably precise EpiXcan is. It flawlessly maps complex diseases right back to their biological source tissues. For schizophrenia, the top tissue contributor for unique associations is brain tissue, at over 30%. For coronary artery disease, it traces right back to arterial tissue at almost 32%. And for lipid traits, like cholesterol, it points directly to the liver at about 26%. So it isn't just finding genes in a random vacuum. It's telling us exactly where in the body those genes are actively causing trouble. And finally, part 5,
repurposing drugs for complex diseases.
This setup perfectly visualizes a computational drug repurposing pipeline. Think about it like this. If EpiXcan tells us that a specific disease turns a crucial gene way up, scientists can then screen a massive library of existing drug profiles. They're basically hunting for a drug that is already known to turn that exact same gene down. By finding a compound that reverses the genetic signature, you effectively reverse the disease state itself.
And the study actually gives us a powerful, concrete example of this in action. EpiXcan was used to analyze the genetic signature of a high body mass index. It then screened for compounds and successfully predicted that an existing one called uracilic acid could reverse that exact genetic signature. The crazy part? Independent literature already reports that uracilic acid increases skeletal muscle and brown fat while actively combating diet-induced obesity. The predictive system absolutely works.
So I want to leave you with this final, pretty provocative thought. What if a simple DNA swab didn't just warn you about your risk for future illnesses, but acted as a perfect decoder ring, telling your doctor exactly which existing, everyday drugs could be used to prevent them before they even start? EpiXcan isn't just a cool data tool. It's laying the very real groundwork for an incredible future of truly personalized medicine. Thanks so much for